So hi, my name is Geelen, and today's question is why high temperature is such a problem for sexual reproduction. And many of you will be aware that plants suffer from heat stress, and this picture of a tomato plant shows you that this can cause severe damage, and in particular with climate change, we expect that there will be much more of these problems occurring in future agriculture. Most of you also probably know that the first damage that happens to the plant is that it will dry out, but it will also show damage to the photosynthetic apparatus. And this is because it damages proteins and lipids that are part of that apparatus. And for example, it will affect the oxygen evolving complex, and that will result in reduced energy metabolism. A major consequence of that is that plants will stop growing, and then it will also cause damage to, for example, development of flowers, and any other process that requires a lot of energy will be stopped or will be damaged. And for example, the production of pollen is one of such processes that require about 10 times more energy than, for example, a regular growth process in the plant. So here is an example of another type of stress that may occur, and that is shown in these two images that are an example of the impact of a heat wave that took place in the summer of 2019 in Belgium in a greenhouse. And what you will notice is that on the left side, it shows you an example of tomato bunch that has been developing under normal temperature. That is that the tomatoes that are closest to the stem are the larger ones, and the ones that are more far away are the smaller ones. And this is because of the developmental process of a bunch in tomatoes, whereby the flowers first emerge close to the stem and then continues to develop more flowers. And therefore you have like a spread in time of developing the flowers, and hence you also have a spread in time of developing of the fruits. In the other example, you see that this situation is reversed. The tomato that is close to the stem is much smaller, and the one that is further away is actually the larger one. So here you may think that, okay, this could be a problem of heat stress that has had an impact on the production of energy, and the energy has gone to the tip of the flower, and or the last, let's say the flower that was developed last. However, that is not what is really happening here. And we can see that because of the development of the sepals. What normally happens is that if that would be the problem, then you would expect to see that the sepals that are closest to, or that are at the fruit that are closest to the stem, that they should also be very small. But this is clearly not the case. And when you look at the arrows, you see that sepals are growing very large despite that the fruit stays small. So clearly this is not a problem of the photosynthesis sink situation where you develop fruits. It is a problem of fertility. So this effect of heat stress on sexual reproduction is in fact something that is more common and reported for most crops. Here are two examples for corn and soybean, where they show that the optimal temperature range for sexual reproduction is always smaller and narrower than what you see for vegetative production. And it's also a few degrees lower than what you have for the vegetative production. So this is a common phenomenon. It's entirely unclear for the moment why plants would have a higher sensitivity during reproduction compared to vegetative production. So this is the question of today. Why is this happening? And perhaps can we also do something about it? Now, before I dive into the reasons why the reproductive system is more sensitive to high temperature, I want to point out that temperature and other environmental conditions are actually complex parameters. And temperature stress, for example, is a function of intensity, duration, and a rate of change of temperature. And it is these combinations of changes and amplitude and length and time that they are applied that can evoke different types of responses by the plant. Now, by a paper from Frachkos-Stefanakis, he shows that in the literature up to 2016, that people have been studying heat stress impact on plants in very different ways. And this complicates the interpretation and the comparison of the data that are out there. He divides 4 types of regimes that are typically applied. We know, for example, that plants are typically highly sensitive when temperature rises above 10 degrees of the normal regular temperature that they grow. And so, for example, a direct applied heat stress of 10 to 15 degrees for a very short time, that will determine a response in the plant, reporting what its basal thermotolerance is. And so this has been studied very often in the beginning, and for example, used for transcriptome studies to see what kind of genes are upregulated when you suddenly raise the temperature to a very high amplitude, and then for a very short time, so that you can then see what the responses of this are. Another type of regime is a pre-induction heat stress, where you pre-treat for a short time with this high temperature, and then look at how the plants are able to respond when you apply a second heat stress for a short time. And this will then measure, for example, the acquired thermotolerance. And the idea was then to see how plants can actually adapt to a change in climate, for example. More recently, we're paying much more attention to the gradual change in rise of temperature. And this has everything to do with the more natural conditions that we expect to occur upon climate change, that in the morning temperature may be low or reasonable, but as the sun rises, temperature increases, and this is in a gradual process, obviously. So gradually increasing the heat stress is another regime that is being applied, and that would then try to process more natural, mimicking natural conditions. And that will also test the acquired thermotolerance. And then a more recent development is to look at mild chronic heat stress, and that will determine the mild thermotolerance. And for example, recent evidence shows that plants can grow longer hypocotyls when temperature rises a few degrees, and that this is connected with an increase in production of auxin. So because of the lack of uniformity of methods that are applied, we need to be very careful when we compare the different results. It can be often be a bit confusing when we look at the different responses that have been reported. Nevertheless, we can divide the impacts of heat stress on different aspects of sexual reproduction. And the first I want to address here is the impact on flower organ development. So what we know is that high temperature can cause early or delayed flowering. It can damage the development of flower organs, and also the synchrony in which male and female reproductive organs are being developed. It can also cause flower abortion, poor dehiscence of the anther, etc. Here I show you 2 examples, one a tomato plant that is sensitive to high temperature that produces elongated pistils, and in an Arabidopsis example where the filament length in the anther is being reduced upon high temperature. And in both cases, you can see that these are plants that show auto-fertilization, they self, that you can imagine that there will be a much reduced fertility because of high temperature stress impact on the development of the pistil in tomato and the filament development in Arabidopsis. However, although this may explain quite frequent infertility reports that have been out there, this cannot be the full story. And it's also unlikely going to impact, for example, reduced fertility in a natural environment, because most plants show they're not selfing, they're actually cross-pollinated. And in this case, you can imagine that insects would still be capable of performing that cross-pollination. A second level where heat can impact fertilization, impact sexual reproduction, is on fertilization. And most of these impacts or these effects are related to pollen, pollen stigma interaction, poor germination, and pollen tube growth. And here you see a few examples of this. So Arabidopsis pollen that is germinated in vitro at different temperatures. You see that the efficiency of germination dramatically drops as temperature goes slightly up. 22 degrees is a good temperature for the temperate Arabidopsis plant, but even just a few degrees higher already shows you a strong reduction in the capacity to germinate. This pollen tube growth or germination can also be seen affected inside the style. This is an example actually from a water stress plant because it beautifully illustrates what can happen. I didn't find a nice example like this one for heat stress, but it has been reported that also heat stress can affect pollen germination and pollen tube growth when pollen are being heat stressed. It actually can also be reduced when the flower organs, the female flower organs, are heat stressed. So in both cases, fertilization. However, most important impact of heat stress is the effects on gamete development. And this is the major topic of my talk here today. Most studied is the male gamete production because of the accessibility of the anthers and isolation of pollen to look at the effects. Ovules have been studied, but these studies are less frequent, and in most cases it's very unclear what kind of defects are really causing the reduced fertility. In the male reproductive system, however, there has been a much better mapping of the different processes that are being affected by heat stress. So in general, we can point to 4 different processes. The tapetum, which is a nursing cell layer surrounding the developing spores and microspores that is responsible for feeding the pollen, is sensitive to high temperature. Then meiosis is very sensitive to temperature, and that we will explain later. And then also microspore development, cell wall components deposition is sensitive to heat stress. And finally, the mature pollen are also sensitive to heat stress. In the case of the tapetum, this sensitivity relates to defects in the tapetum development. For pollen to be developing normally, the tapetum has to undergo apoptosis at a given moment. And in fact, it has to be happening exactly during microspore development. When it happens too early, which is something that can happen upon heat stress, then microspores cannot develop properly. And in fact, what the tapetum does is it delivers energy to the pollen or the microspores, and it delivers also components for cell wall formation. And when it disintegrates prematurely, it cannot feed the pollen with the energy nor with these compounds, and that will abolish any development of the microspores. And often this is then also used in developing completely male sterile plants that are useful for crop breeding. Cold stress can prolong the longevity of the tapetum, and that also has an impact on the pollen production. The sensitivity of the microspore to high temperature may relate actually to the tapetum. That's unclear at the moment. What is clear is that high temperature affects pollen cell wall formation, and probably this is connected to some extent with the development of the tapetum. The sensitivity of the mature pollen relates presumably to damage or repair of proteins and lipids that get unfolded, for example, during heat stress. And because pollen no longer can benefit from external energy sources, they cannot repair that unfolded protein damage. And therefore, they are unable to complete the repair process, and that can inflict reduction in fertility. So that leaves us with the sensitivity of meiosis to heat stress. So why would meiosis be hypersensitive to high temperature? What we know is that the reproduction in eukaryotes, sexual reproduction in eukaryotes, is in general hypersensitive to high temperature. And we know this because there are many examples out there in the animal field that shows that the optimal temperature for example, sperm production is often a few degrees lower than the body temperature. And for example, in humans, sperm production is done in testicles that are put outside of the body, where the system can allow the temperature to be about 2 degrees lower, and just 1 degree higher affects actually the quality of the sperm. So because of this general problem of temperature sensitivity of sperm production, you could argue that this is something that originates far back in evolution of the eukaryotes. So then the question is, when did eukaryotes develop sexual reproduction, and how and why and how did that happen? The current opinions about the development of sex are that it may have been a system that has helped to overcome problems of reactive oxygen species. And this may have been caused by the engulfment of the pre-eukaryote engulfing actually other prokaryotic cells, developing a system of allowing an internal energy provider through the development of mitochondria. And what happened in such a situation that because of the production of energy inside the cell through a dedicated organelle, the mitochondria, a lot more ROS is being produced. And we know that these oxygen species can cause damage to DNA, and that DNA damage repair mechanisms actually respond to increased ROS to repair the DNA. Now, maybe one of the reasons that sex has been developed is that because of this increased ROS, systems of DNA damage repair were not sufficient, and to get some kind of reset of the system, sexual reproduction would allow a much better protection against ROS. And when we look at the system of meiosis and meiotic recombination, these factors are in fact very similar to the DNA repair mechanism that also exists in the vegetative cells. So perhaps the reason why meiotic recombination takes place is because increased ROS production upon high temperature. So when you look at the impact of high temperature on recombination, we see that it will shift the frequency, and it also will shift the position of the recombination. Several studies have been done, and actually it's known for a very long time that recombination is sensitive to high temperature. The way that recombination has been measured or monitored is by looking at the formation of chiasmata. These are structures that can be revealed by performing meiotic spreads. And this is a method by which you take advantage of the large meiotic cell and lyse these cells to allow the DNA or the chromosomes to spread on a glass slide. And here are a few examples shown for barley that produces a chromosome set of 7, 2 times 7 chromosomes that are during the prophase of meiosis pairing. And when chiasmata are formed, they typically form near the telomeric ends of the chromosomes. As a consequence of that, we see the barley chromosomes often as a ring-shaped structure, like shown here in panel A. However, at higher temperature, the position of the chiasmata seems to shift towards more internal positions, and then they are called interstitial chiasmata. And these chiasmata correspond to, in fact, crossovers. And so in panel B, you can see that at higher temperature, the number of ring-shaped structures is reduced, and they are much more rod-shaped type of bivalents, as they are called, which are actually paired homologous chromosomes that are now being connected and a more central part of the chromosomes, allowing them to be visualized as rod shapes. At even higher temperature, the crossover formation of chiasmata formation is being abolished. And in that case, there are other examples here shown on the right in panel N, where the pairing of the 2 homologous chromosomes are actually stopped. And instead of making bivalents, they now make univalents. And this will have an impact on the further progression of meiosis, because that will lead to aneuploidy and actually abortion of further development into a microspore or pollen. Now, in general, meiotic recombination is not only affecting hence the position of recombination, but also the frequency. And this has been reported by 3 research teams in recent years. And the Bombli's lab, they have shown that high temperature actually has an optimum. And when you go below or above an optimum degree or temperature, that you can have an increase in the recombination frequency. In the Copenhagen lab, they have shown that it is actually the type 1 crossover that is highly sensitive to high temperature. And they indicated in their work that at increased temperature, you have an increase in MLH1 foci, which are indicators of the number of crossovers that are being executed. And this is fully dependent on enzymes that execute crossover type 1 crossovers. In my own lab, we have been investigating different positions in the genome of Arabidopsis, and what we found is that not only do we see a change in recombination to a higher number, but you can also have positions where you see a decrease in recombination. And in contrast to what was observed in barley, for example, here we see that the number of interstitial crossovers is actually decreased in this particular position of chromosome 3 in Arabidopsis. So that leaves us with the question, okay, there's change in temperature, but why would, there's a change in recombination frequency, but why would recombination be sensitive to temperature? And this is particularly an interesting question in the light of recent studies that have been investigating factors that would reduce recombination suppression. And these factors are known like FUNC-M, fidgeting, and DNA helicases, REC4AB, that when you mutate these genes, the recombination frequency increases. But this seems to have no impact whatsoever on the fertility of these plants. So apparently, if you increase recombination, that doesn't cause any repercussions for seed set and reproduction. So why would temperature then have to have an optimum? And so this is currently still an open question. And further studies, what people are doing is looking into more specifically which proteins may be sensitive to increased temperature. A theory that has been put out by Bomblies is that at high temperature, there may be a problem of protein aggregations that are involved in development of the synaptonema complex. And this indeed shows in recent papers that a protein that contributes to axis formation during the alignment of the homologous chromosomes is affected by heat. One of those proteins is ZIP1 that will consolidate the interaction of homologous chromosomes. And upon high temperature, the deposition of ZIP1 is being affected. And so this will perhaps influence the chances of recombination knots to be developed. And this could also influence the position as well as the frequency of recombination events. The second type of impact of high temperature on sexual reproduction, and especially in meiosis, is the impact on the cell cycle. Indeed, meiosis requires a specialized cell cycle, and that's because there are two separation events of chromosomes with only one DNA duplication event. So what you have in the normal meiosis is that DNA duplicates during the S phase, and that chromosomes condense and separate during the mitotic event. In meiosis, situation is kind of different. You do have a DNA synthesis in the S phase, but then a process of homologous chromosome alignment occurs and a recombination takes place. And then in the first step, homologous chromosomes are being separated. And instead of doing a DNA synthesis step, this is being skipped. A second cell division takes place during meiosis 2, and that will result in the haploidization of the final cells or the gametes that emerge from such divisions. What we know about this cell cycle progression of meiosis is that it involves a change in the activity of the regulators of the cell cycle machinery, which are typically the CDK cyclin complex regulators. And CDK cyclin interactors or complexes control the process of, for example, cytokinesis. And they, for example, are suppressed they're actually activated during meiosis 1. They should be suppressed when you get cytokinesis. So cytokinesis, for example, may be skipped after meiosis 1 because the CDK-TOM activity is maintained. And that will then cause inhibition of TDM, which will suppress the execution of cytokinesis. And then the second meiosis can take place without a proper cytokinesis event has been established. So when we look at heat stress impact on defects in meiosis, we see that in a number of plants, there are reports that we get formation of diodes. And this is shown in this slide here. In a normal situation, after meiosis, tetrads are formed that are spores that are derived from a single meiotic event that contain haploid DNA and that are still together in a package and wrapped around, encapsulated in a colorless bag. Upon heat stress, we see that in a number of cases, these tetrad stage microspores are actually no longer showing the tetrad configuration, but instead 3 or only 2, and some cases only 1 microspore can be defined. And often what we see is that they contain multiple nuclei. So an example here is where you have a triad with one of the cells containing 2 nuclei, and in another situation where you have a diad whereby each of the spores contain 2 nuclei. And this indicates that, in fact, that regular progression of chromosome segregation events have taken place. However, the cytokinesis process has not been executed properly. In the other panels shows the further progression of the defects upon meiosis. What it actually indicates is that a short heat treatment doesn't stop the further progression of microspore development. So after the heat damage has been done, cytokinesis defects have been accumulated, the microspores continue to develop, and eventually they will form triads, diet, and even tetrad microspores. And so finally they actually develop into viable pollen. So here is another example of the details of the defects in cytokinesis. Currently we know that cytokinesis requires the development of a callose plate separating the individual spores. And upon heat stress, this deposition of callose is being reduced. And in some cases, the cell wall are disrupted, or the position of the cell wall is not correct. And this is probably due to defects in phragmoplast formation before cell wall formation. And examples that are indicated here illustrates that the positioning of the nuclei is not correct. And because of that, the microtubules that form in between the separated nuclei allow them to form cell walls. But in some cases, the nuclei are too close to each other, such that no microtubules are generated between them. And that then presumably causes the formation of a cell that contains 2 nuclei instead of 1. So now when we look back to the impact of heat stress on pollen formation, we can summarize that it has impact on different stages. And here is an example of an experiment whereby we apply heat stress for about 1 day, and then follow the quality of pollen formation in subsequent days. When we isolate the anthers 2 days and 4 days after the heat stress, we see that most of the pollen are dead. And this is presumably because of defects that occur in the tapetum that will disallow these microspores to develop into mature pollen, and that will abolish every further development. When we look at day 5, 6, then we see that there's actually no impact. Apparently the pollen have acquired all the energy they need, and they are at that moment no longer damaged, and they can continue to develop normally. It's only when you look at day 7 to day 9 that some of the pollen appear to be dead, and that's illustrated in this middle panel where you see that there are green and pink stained pollen, and this is because of differential viability staining using the method of Alexander staining that shows that when pollen are actually, when they're pink, then they are viable, and when they are green, they are actually dead. So at this moment, day 7 and 9, that means that when you look at these flowers, that the heat stress they have received is actually right at the moment that meiosis took place. And this relates to the defects that we have just shown in previous slides. And when we look at later time points, then actually these early divisions that are happening before that meiosis takes place, they seem not to be damaging to further development of the anther, and full establishment of anther and microspore development is possible. So that clearly illustrates that indeed the sexual reproduction is more sensitive, that there are 2 more moments specifically that are higher sensitive to high temperature, and that is the development of the tapetum and the meiotic process. A funny consequence of this sensitivity of meiosis to high temperature is that in Arabidopsis accessions that are hypersensitive to high temperature, we can use high temperature treatment to produce pollen that are diploid instead of haploid. And this is shown in two examples here in Arabidopsis accession 1. When you grow these plants at 30 degrees for a short period of time, and then harvest during meiosis, and then harvest the pollen that are derived from this meiotic event, these pollen are by and large diploid compared to the haploid. And this is measured by means of the Coulter counter. Is a device that allows to measure the volume of single cells. Because of the DNA content being higher in diploid pollen, the volume of these pollen is also higher, and they can easily be identified using this machine. In another example, we see Arabidopsis accession 2. You see that there is a small proportion of the pollen that have become diploid. Using the right timing, we can use those pollen and fertilize plants that have been emasculated, and then use the heat-treated pollen to fertilize these emasculated plants. And what we see is that when pollen are derived from a flower that has been harvested 9 days after the heat stress, that we get a reasonable proportion of plants that are triploid compared to what we normally get is never any triploid. So this shows you that indeed high temperature can cause plants to produce fertile diploid pollen. And this may have been one mechanism by which plants have acquired polyploidization. So with that, I would like to prepare a take-home message for you using the symbols. So high temperature causes damage to the photosynthetic apparatus, and this will reduce the energy supply to the plant. And that can have major consequences for flower development. This can have consequences for the production of hormones, distribution of hormones, change in organ development. It can cause the drop of flowers. It can cause the necrosis of flowers because they don't receive the necessary energy. The second level of impact of high temperature is that it will increase the ROS levels, and that plants have adapted to this increased ROS levels by developing repair mechanisms. And one of those repair mechanisms may have been meiosis, and that can then help the plant to overcome this stress that is causing, for example, damage to the DNA, and make the plants more strong and protect them from the future generations from the damage that is being done. But because of this evolutionary development as a response to, for example, high temperature, the meiotic components may still be responding to high temperature, changing, for example, recombination frequency and position. And then a third level of impact of high temperature may be that it influences the cell cycle. And for example, cell cycle regulation by CDK cyclins may be affected by high temperature, changing, for example, the speed of development of the tapetum compared to the speed of development of meiosis and microspores. It may affect also the progression of meiosis, for example, leading to changes in cytokinesis. And when that happens, it can influence segregation problems, but may also in some cases be beneficial to the plant, allowing, for example, polyploidization. With that, I would like to finish my presentation and hope you have enjoyed it. Thank you very much.